This is a strand of DNA. It is composed of two anti-parallel strands oriented in a helix shape. It has both a 5' and 3' end. For transcription, the template strand is used. This is because if the coding strand was used, it would result in a different protein since RNA is always synthesized in the 3' to 5' direction. We will now take a closer look at the nitrogenous bases. Every DNA strand is composed of the same 4 nitrogenous bases. The nitrogenous base adenine is a purine, meaning it has 2 carbon nitrogen rings instead of 1, making it bigger than thymine, its complementary base pairing. Thymine is a pyrimidine, meaning it has a single carbon nitrogen ring opposed to 2, and is smaller than its complementary base pairing adenine. Guanine, like adenine, is a purine, and its complementary base pairing is cytosine. Cytosine, like thymine, is a pyrimidine, and its complementary base pairing is guanine. Held together by hydrogen, also known as H-bonds, adenine and thymine have 2 H-bonds holding them together, making them easier to break than guanine and cytosine, who have 3 H-bonds between the two of them. Helicase is an essential enzyme in transcription. It unwinds the DNA strands, separating them by breaking the H-bonds between the complementary base pairs. Here we can see helicase unwinding the DNA strand. RNA polymerase is an enzyme. It is responsible for transcribing the template strand on the DNA into an RNA sequence. It does so by matching the complementary base pairings on the template strand. RNA polymerase is used solely for transcription, and it is not involved in the replication of DNA, as that would be DNA polymerase. Here we can see RNA polymerase synthesizing the new mRNA strand. It does so in the 5' to 3' direction, and new nucleotides are added in the 3' direction. As RNA polymerase goes along the template strand, it will add uracil instead of thymine, so whenever an adenine is read for the complementary base pairing, instead of thymine, uracil is put in its place. This is because uracil is more easily broken down than thymine. This combined with RNA's ability to degrade quite easily makes it the better option for synthesis on the RNA strand. After transcription, the pre-mRNA strand must be edited. This is because certain regions on the mRNA strand will not code for the protein. A protein complex, known as a spliceosome, will remove the introns and splice together the exons. Introns are long sequences of nucleotides that do not code for a specific protein. Instead, they interrupt the sequence of genes and therefore must be removed. Here we can see the spliceosome splicing together the exons. After splicing, exons are what remain. This is what will form the mature mRNA. They are the specific set of nucleotides that will code for the protein. To one end, a 5' cap is added, and a poly A tail is added to the 3' end. Together, they ensure the mRNA strand will not degrade before it reaches the ribosome. After processing, the mature mRNA strand can now leave the nucleus. It travels through the cytoplasm to reach a ribosome. A ribosome is an organelle composed of 2 subunits, the small and large subunit. These separate units join together on the mRNA molecule. A ribosome has 3 active sites, an amino site, a peptidyl site, and an exit site. A tRNA molecule will now join the P site carrying the amino acid methionine. Transfer RNA is a single-stranded RNA molecule with an anticodon sequence found at the bottom-most hairpin-like loop. This anticodon sequence will complement the codon triplets on the mRNA strand, bringing the appropriate amino acid to the site of protein synthesis. Here we can see tRNA with the anticodon sequence to the start codon AUG, bringing that corresponding amino acid. A second tRNA molecule will now join the A site. The mRNA strand will now shift one codon sequence over, causing the tRNA molecules to follow. Here, the tRNA's anticodon sequence will match with the codon sequence found on the mRNA strand. A peptide bond will now form between the amino acids, marking the start of our polypeptide. The tRNA molecule will now leave through the E site, while a new tRNA molecule joins the A site. The process repeats until a sufficient amount of amino acids have joined the polypeptide. Termination begins when a stop codon is read. A protein-releasing factor will bind to any ribosome with a stop codon. There are 3 stop codons. A protein-releasing factor will float in from the cytoplasm to join the A site of the large subunit. It will cause hydrolysis to take place on the tRNA molecule, releasing the completed polypeptide from the ribosome. The site of protein synthesis is then deconstructed and will be recycled by the cell.